Hi, I'm Alice Law, a Senior Application Scientist with NanoFCM, and today I will be introducing you to our nano flow cytometry platform, the Nanoanalyzer, and highlighting publications where the Nanoanalyzer has brought speed and certainty to physical and biochemical analysis of EVs. So this presentation will focus on EVs, but I do want to highlight that our Nanoanalyzer platform has been built for the dedicated purpose of characterizing biological and inorganic nanoparticles from across multiple fields, including virology and nanomedicine. So as a company, NanoFCM aims to bring reliable and quantitative measurements to all aspects of the nanoscale. This is in order to support the emergence of new classes of diagnostics and therapeutics. So before delving into our nano flow cytometry platform, I wanted to start with an overview of different technologies applicable to nanoparticle analysis. This will help highlight the need for a platform and the position that it fills. On this slide, I'm sure you can identify a number of bulk analysis techniques such as laser, immunoblotting, and dynamic light scattering. However, to keep pace with the direction that modern therapeutics and diagnostics are heading, bulk analysis techniques are not enough. There is a single particle analysis is essential to answer crucial questions about your particles. For investigation into the physical properties of nanoparticles on an individual level, researchers have typically turned to nanoparticle tracking analysis, which sizes particles and provides concentration measurements based on their Brownian motion in suspension. So despite its ease of use, there is some dispute over the accuracy and reproducibility of these measurements. TEM, however, provides a high level of sensitivity and increased resolution in comparison to NTA. Unfortunately, it is a time-consuming technique, needing a high level of expertise to perform. Resistive pole sensing is another classical technique, which measures the changes in current across a small pore as individuals pass through, providing size and concentration measurements. Similar to TEM, RPS provides a high level of sensitivity, but it too suffers from very slow analysis times due to samples interacting with the pore and causing blockages. The thing is, these techniques only inform us of the particle size and concentration, the physical characteristics. They fail to provide valuable information on the phenotypic properties of the particles, meaning that researchers are often forced to use multiple systems to achieve a clear picture of their particles. Fluorescent NTA perhaps comes closest to being a comprehensive platform for physical and biochemical analysis, but its sensitivity and accuracy is lacking. Flow cytometry holds a huge amount of potential for EV characterization. However, again, conventional cytometers lack that resolution and sensitivity needed for nanoparticle analysis. So in the last slide, I mentioned some of the crucial elements needed for nanoparticle analysis, including single particle detection, physical and biochemical characterization, as well as a high level of sensitivity and resolution. Ideally, these features would all be provided in one comprehensive platform, reducing the need for multiple techniques and increasing data throughput. So from here on out, I hope to show you how our dedicated nanoflow cytometry platform, the Nanoanalyzer, fulfills the criteria on this slide and your expectations for comprehensive technology. So one of the reasons that we think nanoflow cytometry will become increasingly relied upon as a comprehensive EV analytic technique is that flow cytometry is already widely used and a very much robust method. So for those new to flow cytometry works by passing particles through a laser. Light refracted off particles is detected and then informs us about the presence of the particle and its size. Coloured light emitted by fluorescent probes conjugated to antibodies provide information on things such as surface proteins. So since its conception, flow cytometry has shown itself to be a versatile technique and a premier tool in single-cell analysis. However, conventional flow cytometers struggle to provide the sensitivity requirements for the analysis of nano-sized particles such as EVs, with light scattering detection limits of approximately 200 to 500 nanometers, dependent on the refractive index of the particle. So to facilitate identification of these nanoparticles, fluorescent staining is often used to resolve the particles from the background. However, fluorescent signals from nanoscale particles will be orders of magnitude dimmer than that of cells and may be beyond the fluorescent detection limits of conventional flow cytometers. Additionally, this detection method may lead to the inclusion of false positives such as loose fluorophores or degraded membrane. The Nanoanalyzer extends the applicability and versatility of flow cytometry to nanoscale particles, unimpeded by sensitivity and resolution limits of the conventional flow cytometers. But how does it actually achieve this? Well, the enhanced sensitivity comes from a number of factors. So in the Nanoanalyzer, the sample fluid is hydrodynamically focused to a very fine stream, approximately 1.4 micrometers, via the gravitationally fed sheath fluid, and this ensures individual particle measurement and prevents swarming from happening. So the transit time of the nanoparticles passing through the laser is extended to milliseconds, and this leads to a longer dwell time and increased photon generation for detection by our detectors. And finally, the Nanoanalyzer utilizes highly sensitive single photon counting modules for side scatter and fluorescent detection. And this ensures enhanced quantum efficiency of photon detection, which just makes it ideal for EV research. Another thing that sets the Nanoanalyzer apart from other flow cytometry-based techniques is the simultaneous measurement of both side scatter and fluorescence. This means that false positive signals generated by loose fluorophores or things such as degraded membranes are not included in the analysis. So on this slide, you can see the burst event traces of a stained EV sample, whereby the blue side scatter burst event traces indicate a particle, with the signal intensity reflecting the particle size. So the green event burst traces, these are measured simultaneously and indicate which of these particles carry the stained target. So on the right, you can see the dot plot, which is generated from the detection of these particles, utilizing the side scatter channel as the triggering channel, which means that only particles greater than 40 nanometers are shown on this dot plot before separation into the fluorescent positive and negative populations on the y-axis. Any unbound fluorescent signals not associated with the side scatter signal, none of these are included on the dot plot, and this really ensures robust and true data. So with one side scatter detector and two fluorescent detectors, the Nanoanalyzer can perform label-free analysis to provide size and concentration measurements of individual particles, and fluorescent labeling is also possible. Working with fluorescence can be very flexible with both single and dual staining of external proteins, lipids, and nucleic acids possible. And this really allows individuals to identify and characterize EV subpopulations with their samples very easily. So going on to kind of the Nanoanalyzer performance now. So the Nanoanalyzer provides sufficient sensitivity to analyze biological particles down to 40 nanometers, with sizing resolution comparable to that of TEM. Smaller diameters can be measured with inorganic particles, and this is due to their often higher refractive properties. And then you can also see in that bottom left panel there that detection of single fluorophore molecules such as PE is possible. And this really opens up avenues for the detection of rare markers on your EVs. So a further look at how the Nanoanalyzer measures up against other technologies with regards to physical characterization of EVs. So in that top panel there, the Nanoanalyzer compares favorably to cryo-TEM, and this highlights that heterogeneous EV samples can be accurately measured by nanoflow cytometry to comparable levels to TEM, but at much greatly improved analysis times. In the bottom left panel, you can see that the Nanoanalyzer and RPS systems return very similar profiles with a peak around 60 nanometers. However, the NTA shows a much broader size distribution and higher peak. This broader size distribution and large peak with NTA measurements is also evident in that last example there. And this example shows particle size distributions of EV preps separated by different methods. So in comparison to NTA, the Nanoanalyzer had a much smaller size range of 40 to 150 nanometers in this example, and peaks of about 60 nanometers versus 110. So the data generated using our software is then translated into a final simple data report. And this just takes around 2 to 3 minutes to do. So the final report gives us detailed information on particle concentration, which is given in particles per mil, and takes into account any dilutions and false positives from blanks or buffers you may have applied. And then we have size distribution, and here you can have full control over the x-axis, allowing you to examine specific particle sizes of interest. And then finally, we have the fluorescent population, where you can use gating to identify fluorescent star populations and their associated concentration and size distribution. So the collected data can also be saved in a number of ways for external analysis in other software, such as FCS files for use on Flojo. So here I just want to highlight the workflows a user can expect with the Nanoanalyzer. For daily setup of the instrument, fluorescent quality control beads are run to align the system to allow comparison between individual runs over multiple days. These QC beads also act as a concentration standard. And then a silicon nanosphere cocktail is used as a size reference standard, and this is due to silica's similar refractive index to EVs. So from here, a standard curve is constructed from the particle sizes and their side scattering intensity. So there is a huge amount of flexibility in the use of the Nanoanalyzer, with both label-free analysis and fluorescent labeling available from biofluids and conditioned media. So if the staining procedure has been optimized, then often the removal of free dye is not always required for fluorescent analysis, making this a very quick workflow once optimized. So the system is high throughput, with each sample only taking 1 minute to acquire and 2 to 3 minutes to analyze. So hopefully over these past few slides, I've shown you how nanoflow cytometry and the Nanoanalyzer can easily compete with other nanoparticle analysis techniques out there. And so now we're going to move on to some data to show you how the Nanoanalyzer can be applied to enhance EV-related research. So the Nanoanalyzer's unique sensitivity at size scatter detection in this example provides a quick and comprehensive comparison between EV isolation methods. In this 2020 JEV article, the size distribution and concentrations of EVs isolated by 6 different methods were assessed using label-free analysis. Of these, you can see in graph B, ultra-centrifugation stands out with a unique broader size distribution compared to the other methods. However, if you look to graph H, you can see this is actually with a much lower particle count. So the authors related this to differences in purity between the preps, with non-EV particles still present after most EV isolation methods. So using detergent, the authors were able to confirm the physical nature of particles versus non-EV contaminants such as lipoproteins. So you can clearly see the ablation of particles following treatment in both the event burst traces in A and between the counts in B and C there. So using this detergent method, the authors found that the EV preparation by ultracentrifugation showed a much greater purity than that of commercial kits, providing kind of insight into the impact of different isolation methods in a very quick time frame. So these slides are a great example of how high resolution side scatter data can inform us of the total particle information, but it's really the addition of fluorescence that can highlight and characterize the true EV population within complex samples. So this is an in-house example of antibody tagging in a complex mixture to pick out particles that may overlap in size distribution. So here, EVs were stained with a FTSE conjugated antibody, which allowed us to generate 3 different panels. First of all, we have the total particles, the FTSE positive EVs, and then the FTSE negative EVs. So you can see in the total population, there is 2 peaks in the size histogram, with peaks around 55 nanometers and 85 nanometers. So you can see that our fluorescent EVs are all part of this larger size EV population. But then if you look to that bottom panel, you can see whilst all fluorescent EVs are large, not all large EVs are fluorescent. And you can clearly see 2 size distributions in the FTSE negative population. So taking this use of fluorescence back to published data, Here we have an experiment where nanoflow cytometry was used to track changes in EV size distribution and the presence of aquaporin 4 following electrical stimulation. So in the top there, you can see the size distribution and dot plot of unstimulated astrocyte EVs. And in that dot plot, you can clearly see the presence of 3 different populations. So first of all, we have the small aquaporin negative EVs in the bottom left, the small but aquaporin positive EVs above this, and then on the top right panel, the larger aquaporin positive EVs. So when low electrical stimulation was applied, in this case 2 Hz, you can see that there was a shift in the size of these vesicles as well as the expression of aquaporin. And then with this increasing electrical stimulation, you can see that depending on this level of stimulation, the EV profile is very different in regards to the expression of aquaporin 4 and also the size distribution. So this is just a really nice way of capturing EV outputs using nanoflow cytometry, and I would recommend checking out this paper. So here you can see the use of immunofluorescent labeling to assess CD147 positive EVs. So CD147 is overexpressed in some cancers and can be used as a biomarker for colorectal cancer. So in the top panel, you can see the dot plots of CD147 fluorescence versus side scatter for EVs isolated from colon fibroblast cells and cancer cell lines. So it's worth reiterating that all these data points on the dot plots will have passed our side scatter threshold, determining them as a particle over 40 nanometers. So in the fibroblast cell lines, less than 10% of the EV population was CD147 positive, whereas this proportion increased to nearly 50% in the cancer cell lines. And you can also see this is in very much agreement with the Western blot. So as the CD147 levels differed among normal and cancerous cell lines, the authors then sought to determine if CD147 levels could be used to differentiate between EVs isolated from plasma of cancer patients and that of healthy donors. So in the bottom panel, on the left, you can see a representative dot plot of a patient, and on the right, the significantly higher particle concentrations of CD147 positive EVs in cancer patients. So this is just a really great example of to demonstrate the flexibility of the Nanoanalyzer to handle both EVs isolated from conditioned media and biofluid for investigating novel diagnostics. So the past few examples have shown single staining. However, the Nanoanalyzer is fully capable of detecting dual staining as well, using its 2 fluorescent detectors. So here, this example, you'll be able to see the use of dual staining to identify a specific EV subpopulation. So in the blue box there, you can see the total number of particles above 40 nanometers, and this is based on the side scatter detection. From these particles, total particles, we can identify an EV population based on the FITC positive lipid dye staining, and this amounts to around 80% of the total particles. And finally, we can now gate the double positive population to identify the subpopulation displaying our protein of interest, which is conjugated to APC there. So this is a really nice and easy example of how you can use dual staining to really zone in on those EV populations that you are looking for. So the next couple of slides show some examples of dual staining in published literature. And to start off with something slightly different, we're going to be looking at utilising EV components to deliver adenovirus. So oncolytic adenoviruses, whilst having strong cancer-destroying potential, often suffer from degradation and removal by host immune responses. Encapsulating the virus in an EV membrane may allow for avoidance of these responses, ensuring effective delivery to cancerous sites. In this paper, 2 different stains were used to evaluate the encapsulation efficiency. So first of all, we have the EV marker, CFSE, a membrane permeable dye activated by S races found in the EVs, and then the viral marker, a cytodye for the viral genome. So this staining was used, the concurrent staining was used to look for the oncolytic adenovirus encapsulated EVs, which would show concurrent CFSE and cytostaining. And you can see there, it's highlighted and then it amounted to around 60% of the particles. So using this staining method, it was also possible to detect the empty EVs devoid of the virus by CFSE staining alone, and also the unencapsulated virus by cytostaining alone. So again, it's just worth reiterating that all the data points on this dot plot will have passed our size getter threshold, determining them as a particle over 40 nanometers. So coming to our last example now. In this paper, the authors, including our very own Ben Peacock, investigated whether neural stem cells release and traffic mitochondria via EVs to restore mitochondrial function in target cells. So mitochondrial dysfunction is an established feature of several inflammatory and degenerative neurological disorders. So here, the Nanoanalyzer was used to perform biophysical characterization of the EVs. However, the data you see displayed here may be slightly different from that you'll see in the paper, as it's been generated by our own software. So here, dual staining was used to identify whether these EVs actually harbored mitochondria. So CD63 was used as a general EV marker, and MitoTracker red was used to label any mitochondria present. So the authors found that among the total EV population, around 35% of EVs sized between 150 to 800 nanometers were positive for MitoTracker red, whilst only a small percentage of the smaller EVs sized between 40 and 150 nanometers were positive for this MitoTracker red. So this suggests that the larger EVs are capable of harboring intact mitochondria. So this really is an exciting find that actually might pave the way to the development of novel EV-associated approaches to restore mitochondrial dysfunction in degenerative neurological diseases. So coming to an end of the presentation now, so I'd just like to say thank you for listening. And I hope that I've managed to demonstrate how the Nanoanalyzer can be used to perform rapid and comprehensive characterization in multiple areas of EV research. So again, thanks for listening, and please go and check out our NanoFCM booth to further explore the capabilities of the Nanoanalyzer and to find out how nanoflow cytometry can be used to achieve your research goals and strengthen your data outputs. Thank you again.